Anselm, what are you actually printing here?
What we're trying to do is to make genes that are going to be used by researchers studying stem cells, studying how cells change and how they work. We're making genes for people that are actually engineering microbes to make chemicals and to replace like petroleum-derived chemical feedstocks.
How do you feel about GMOs? What are the perils and where are the positive aspects?
Well, I'd say for the most part, actually, most of the modified organisms are not actually in nature. They're typically found inside labs or maybe some small factories. The one big exception, of course, are the agricultural, the plants that have been engineered to have certain resistance genes. I think the real challenge there is not so much environmental, because frankly most crop plants don't survive on their own in the wild if humans aren't tending to them. Even the GMO plants are pretty weak when it comes to natural selection in competition with their wild cousins. But the real issue is with GMOs is around, I'd say, the ownership of the food supply and the ownership of the biological commons. So for me personally, I think I'm most concerned with how a very old, centuries-old body of patent law is interacting with these radical new inventions, and how it's sort of enabling, I think, the private sphere to encircle, to own the commons, the biological commons that is sort of our ancient birthright. I'm just a bit worried that there's not enough concern for what I think the really deep issues are, which is around how and who can own things that have been with us for 4 billion years.
Now that the genome has been sequenced, how close are you to printing organs and more complex living beings?
Complex living creatures is probably a ways off, but there are early inroads in making organs, and people have begun making bladders, say, which is one of the simplest organs in the human body. And I think there will be a lot of progress in this over the next few decades, and there's a lot that I think these genes, these synthetic genes, can do to help us, either indirectly encouraging the cells to form full organs, but more importantly probably just to help us reverse engineer how cells actually develop into these complicated structures. So we're at a point where we have the sequences, we have the full genome sequence for the human, and we begin to sort of understand what all the pieces are, but we don't yet understand how they all interact to form these complicated functions, how they read information from their environments and make decisions, and really how does the circuitry of life work. We're still at a very primitive stage when it comes to understanding it. And one of the key things you have to do to really understand a system is to be able to tweak it and change it so you can begin to understand how the pieces work together and really understand how they work together as a system. One of the big pushes in synthetic biology is actually just to use a sort of constructivist approach to understand how biology works.